Good
Hey, good
morning. Can
morning,
you?
Valley.
I
Yes, I
cannot
can
hear.
hear.
Let me
I,
see. Mm, Can you hear me?
yes.
Yes, now I can hear you as well.
Okay.
Perfect.
Good morning, Valley.
Good
Thanks,
morning.
thanks for give the time to us for your exciting talk. We really
Yeah.
look forward to this talk.
Okay, thank you. Let me see. Will I? Oh, yeah. No, okay. So I will be able to share screen later?
Yeah, I think you can try now and see if it works.
Okay, let me see. First, I have to. Have to see which screen is best to share. Let's see this maybe. And then, is this okay, or I should share the small screen?
I think it's perfect. It looks perfect.
Yes.
Yeah.
Okay. Yeah. Then I will do it like this. But maybe now I should stop sharing because you had also something, I guess. There is some noise. I don't know.
There's voice noise.
Noise. I don't know. Maybe
Uh,
it's some background noise.
I can. I think it's because of my computer. I can mute it when your talk starts.
Okay.
Yeah. So let's try. I just mute myself and see if you can still hear the noise.
Yes.
Sorry about that.
It was just the computer. Okay. No
Yeah.
problem. Yes.
So we just wait for 10 more minutes and we start officially. And you see, we only have few people here. It's not that we have only few audience. It's because we join as a panel.
Yes.
That's why. So, yeah,
Yeah, because it's also on a YouTube channel or yeah.
yeah, YouTube channel, and I think there's another platform, Chinese platform. So there will be audience from all over the place.
Okay. Yes.
Yeah.
Yes. There might be someone joining also that I sent the link to.
Yeah.
Who follow my talk.
Yes.
So
Always is welcome. And I followed your account, social media account, and also I tweeted this in my
okay.
Twitter account, and the EEH the journal also tweeted in their official account in Twitter.
Yeah.
So we might have some big audience today.
Oi, oi, oi.
Yeah.
No, Junji my postdoc Junji, he forwarded me something about I think the release from the journal. Yeah.
Yeah. Yeah.
Which was partly in Chinese, so I couldn't read it, but
Yeah. Yeah. So there might be some questions in Chinese. Then I will just translate to you.
Please.
Yeah. Yeah. So Valley, you are now in Norway or Belgium?
In Norway,
Norway?
yes.
Norway.
Yes.
Okay.
Yeah. Mm.
Okay.
Yeah. Because I am Belgian, but I moved to Norway like 9 years ago.
Yeah. Yeah.
Yeah.
I follow your research, although I don't like know you in person, but I read many of your bird
Oh,
bird
that's
papers.
nice.
Yeah.
What are you specifically working on?
I'm an aquatic toxicologist, so fish.
Yes, fish. Yeah, we have
Yes.
some people working here on fish as well. So
Yeah.
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 Mm.
I think our field is very similar, just different animals, uh, wild. Oh, so hi, hi, doctor. Okay, you muted. We cannot hear you. 唐老师，我听不见你说话，你那个可能静音了。
Oh, sorry, sorry. Hello, Miss Jasper, Professor.
Hello, nice to meet you.
Welcome. Nice to meet you. Hello, Mr. Professor Xu.
Hi, nice to meet you.
So where are you? Where are you from?
We are in Nanjing. In Nanjing, in China.
Okay. And you're there now.
Yeah.
Okay.
Welcome.
different time zones also, yes.
Yeah,
For me, it's in the morning.
yeah, but we are the same. We're the same valley. I'm in Denmark, so we're
Yeah. I saw that you are in Denmark. Are you at Aarhus or somewhere else?
Sudansk University, so
Oh, I don't know where it is.
in Odense, University of Southern Denmark.
Okay. Yeah.
Yeah.
We are in the afternoon now.
Yes.
And Valley, our EEH journal is a relatively new journal. It just launched recently, but we cover a lot of environmental topics, including diversity, ecotoxicology, a lot of exciting environmental field topics. And we are inviting a lot of the renowned researchers like you to give us talks, cover those topics. I think you are number 12 or 20 already, so we already invited a lot of professors globally.
How often you do this webinar?
So far, I think it's twice a week, right? Professor Tang, is it twice a week now?
Yeah.
2周一次吗？现在 Yeah.
Let's just talk.
Yeah.
This is the 14th.
14th. Okay.
Yeah, 14th.
So very busy.
Yeah.
Alvis, did you want to share your screen before, or should I share my screen again?
I think you just start with yours. I don't have anything
because
to
you
share.
had something on the when
background.
we started, so I was not sure if you wanted to share your screen first.
I don't think so. You can just start with yours.
Okay, then I will already do that. This. This.
唐老师，咱们通常是问题是在报告结束以后来提，对吧？中间不提是吧？
那个徐老师，我们这样的，我们有2个直播平台，里面可能会有一些问题的，然后我们的编辑会发到这个 chat 聊天室里面，到时候您可以让我们的这个主讲嘉宾看着回答，如果中文的话可能会翻译一下，到时候。
好的哦 ，OK。
OK.
So Valley, because we have multiple platform, and the questions will be all transferred to the chat here, and both of us can see it in the chat. But you won't. But we will only do it after your talk. So don't worry about during the talk.
Okay. Yes, because I cannot not look and read all the time. Yes.
No. Yeah. No worries.
徐老师，我要不要共享一下那个我们的那个 EEH 那个宣传片？
啊，可以。
哦，好像那个Jasper 他已经共享了，可能
Okay.
我先
Valley, so Professor Tang would like to share a video about our journal. Would you stop sharing for now? And then
Yes.
she gonna play the very nice video about our journal.
Oh, I have to. I try to stop sharing, but it's not. What is this?
能看到吗？
可以。
Health is an eternal topic for mankind. We are born on Earth, and our health is intimately linked to the environment and ecology. The environment is the basis for human survival and development, and every subtle change in the environment will eventually have an impact on biodiversity, ecosystem, and our health. Exploring how the environment and ecology affect our health in the past, present, and future is the mission of Eco-Environment&Health. Eco-Environment&Health is a journal initiated by prestigious scientists with a strong editorial board including multidisciplinary scientists from all over the world. With the support from EEH, you will have a better publication experience. EEH has its own offices of publishing, language, design and broadcasting. The experts will accompany you like an harmonious band and spare no effort to sublimate your valuable scientific research into a magnificent and impactful masterpiece. We have a dedicated center for decision making that deeply integrates informatics, ecology and environmental science. It explores global research hotspots, predicts discipline trends, monitors the influence and dissemination of publications, and supports the journal's smart decision-making. Upon receiving your submission, our assistant editors will contact you immediately and keep you informed of the manuscript status throughout the entire process. The 14-day rapid peer review will make the publication of your research one step ahead. Your distinguished work is worthy of being known worldwide. We will promote your research globally in a three-dimensional manner through various social media platforms. In addition, the open access fee will be waived for the first 3 years, making your research easier to be read and cited by your peers. We continue to explore and innovate the service strategy of the journal. EEH leads ecology and environment to a healthy future.
好了 ，Welcome。
Thank you, Professor Tang. So I think it's time. So why don't we start now? Hi, good morning and good afternoon, everyone. So this is one of our EH talk. So today it's my pleasure to introduce Professor Veli Jaspers, a full professor in environmental toxicology at the Norwegian University of Science and Technology. And she's currently the deputy for the head of research at the Department of Biology and part of the lead group. She teaches classes in the International Master Program, Environmental Toxicology and Chemistry at the NTNU. Further, she is course leader for the courses Ecotoxicology and Practices in Ecotoxicology in the third bachelor of the biology program. And her research aims to understand the effect of pollution and multiple other stresses on individuals and populations. And most of the projects under the umbrella of their group focus on bird species, including songbirds, seabirds, shore birds, and raptors. but studies on rodent and Daphnia are also performed. Studies are carried out both in vitro, so to elucidate the cellular responses to stressors, and in vivo, ranging from laboratory experiment to field studies on wild birds. She has published very impressive over 100 journal papers and received over around 4,000 citations. And we're so excited to have her here today. So Valley, the Zoom stage is yours.
Thank you very much for the nice introduction. And also thank you for the organizers to invite me for this webinar. So today, yes, I will talk mostly about birds, as is already been mentioned. I work mostly on birds, and I will talk about them in a multiple stressor context, and especially also in relation to pollution. So for this talk, I will first introduce the field of bird ecotoxicology. Then I will talk about multiple contaminants, so the interplay between multiple contaminants. And we have done some studies using some mixture exposure experiments. Then I will go to multiple stressor studies and environmental factors using wild birds in the field. And lastly, I will talk about the interplay between pollutants and infectious disease in birds, specifically avian influenza. So for the introduction, the field of bird ecotoxicology is actually like very much linked to the field or the start of ecotoxicology in general, because after World War II, there was a lot of use of many pesticides and other industrial chemicals, basically. And one of them was DDT. And as you can see, DDT was also advertised for use both in the house and around, even with these sort of commercials. Now, this massive use of industrial chemicals led to declines of many bird species. And this is very well described in a book of Derek Radcliffe from 1980 on the peregrine falcon. And so this figure is in fact one example of a species, hence the peregrine falcon, that has been declining during that period. So from, let's say, 1950, 1960s until 1980. And so what you can see on this graph is the United Kingdom. And on the left-hand side, you see the percentage of tiled land, so agricultural lands in the United Kingdom at that time. And on the other side, you can see the zones where the peregrine falcon is like absent or more abundant. So the darker the zone, the more abundant was the peregrine falcon, and the lighter it was absent. So what you can see is that these two maps are almost opposite of each other. And so where the agriculture areas were very high, there was a almost absence of peregrine falcon, and where there was no agriculture, the peregrine falcon was very abundant. So showing the clear link of the use of these pesticides and the decline of the peregrine falcon. So this was actually the launch of ecotoxicology as we know it, because this was the first time that people were also asking questions about, okay, so things that we are producing and that we are releasing in our environment may actually have a harmful effect. And that's how people started studying more about these compounds. So in many of the studies in the beginning, they were using raptors. And raptors are, of course, very beautiful creatures, and many people like working with them, and also they get a lot of attention by the public. But raptors also can be very difficult to work with. As you can see here, just to get a sample from a raptor species, this is a northern goshawk nest, you need to climb a tree. So this means that it takes a lot of time, and you have to drive from one nest to the next, and it poses a lot of challenges. So one can ask the question, why should we use birds? So of course, there's the possibility to not just only use raptors. There are also other alternatives like small passerine birds, or like also seabirds, as you can see here. But in general, birds show a high accumulation of pollutants because most bird species are high on the food chain. So of course, raptors are on the top of the food chain, and that's why they can have these very high levels. but also other birds. They generally feed on maybe fish, or they can feed on insects, so they may show some accumulation of pollutants, and therefore we also may expect to have effects in those species earlier because they have a higher pollutant load. We find birds actually everywhere in most habitats, both in aquatic, marine, terrestrial food chains. So that means that we can also compare different habitats and also different areas around the globe, and also depending on what type of diet they are eating. And like I said, birds, we can almost find them anywhere. So some bird species are actually globally distributed, and then we can study geographical patterns, so which is very interesting. And before we even talk about ecotoxicology, birds were very highly studied in terms of ecology, in terms of behavior, in terms of physiology. So there's a lot of biological background information you actually need to interpret the results. So it's very highly studied species, and also many volunteers are involved in looking at birds because the public likes birds. So like I said before, in the past, monitoring of pollutant levels in birds has focused on these industrial chemicals that were used originally. So, and most of them, we can classify it as persistent organic pollutants. So they are regulated now. And these persistent organic pollutants were mostly organic compounds with chlorine atoms. So among those, DDT, the pesticide, but also PCBs, dioxins, and other organochlorinated pesticides all have chlorines on their organic structure and are regulated now as POPs. So those were very toxic compounds, and that's also why they have been regulated now. And this is just another example of some seabirds in the Great Lakes in Northern America. And there, the lakes, the industry that was closely related to the lakes just discharged all of the leftover chemicals into the lakes. Just thinking that it was such a vast amount of water, which is really true, it's like more like a sea, and that it would just dilute and not have any effects. But as you could see, they had many effects on different birds living there. So they had deformities like crossed bills, so the birds cannot feed, so they will die, and also many deformities in the embryos, like missing limbs and other problems in the embryos. So this also led to massive declines of seabirds in the Great Lakes. And this was one of these other cases where it became very obvious that we cannot just release any compounds into the environment. So past monitoring activities were mostly on these persistent organic pollutants, and they have been regulated now. So, and that has shown to have very positive effects because many of the populations have actually recovered. And for example, the peregrine falcon has returned in countries in Europe that it was completely gone. So this recovery of populations is an indication that it's become better. Of course, chemical industry had to look for some replacement compounds. And so those chemicals that they are producing now, we can call them emerging compounds. And some of these compounds I can list here is, for example, flame retardants that are used for fire safety in many products, and also perfluorinated chemicals like PFOS that you may have heard about, that are also used a lot, both as fire extinguishers, but also like surfactants to like protective clothing, maybe Teflon pans, so things that like kind of make sure that are water and lipids repellent, basically. So the focus has shifted now also in bird ecotoxicology more to these new pollutants and to mixtures of these new pollutants with the old pollutants, because these persistent organic pollutants, they are so persistent that they are still around everywhere. So we can still measure them in high levels in the birds. So we look now at chronic low-level contamination and effects of both old and new pollutants, and also focus more on interaction of these pollutants with other stresses that the birds face in their environment. Just think about global change. So this has a lot of effects on the habitats and on the diets that the birds are exposed to, and in addition, they are exposed to these pollutants. So this is the focus of the field right now, and also will be more the focus of my talk. So the first project that I was involved in is the new Raptor project. So when I started here at NTNU in Trondheim 9 years ago, almost immediately after I got funded this project. And so I will talk a lot about this because I have a lot of results on that. So in this project, we did laboratory studies, so meaning controlled studies of exposure of birds. And there we wanted to see if bird embryos can biotransform some of the emerging contaminants, because we did not know. And also looking how they could affect the embryos. Then this we did with egg injection experiments. And then on the other hand, we did field studies with raptor species, being the northern goshawk and the white-tailed eagle. And we looked if those birds were exposed to emerging contaminants as well as POPs, and also if these contaminants had an effect on their health status. And those samples that were obtained were non-destructive, because raptor species, they are protected species. So we have to think about which samples to take. So just to mention that this work was done by a whole bunch of people, so just presenting on behalf of everyone, and many collaborators and also many students were involved. So that will bring me to the next point about the mixture exposure experiments that we performed within New Raptor. So because of many legislative and ethical considerations when working with birds and especially raptors, we cannot do experimental studies on them. It's very difficult. There's only in some cases that experimental exposure studies can be done on raptors if these raptors have been kept, for example, in generations already in the lab. So kept and bred in the lab, basically, or in closed experiments. So we used as model species, and those model species were the Japanese quail and the chicken. And we did 2 exposure experiments. So on the chicken, we used flame retardants that we injected in the egg. And for the chicken, we used PFAS that we injected in the egg. And then we looked at the transfer. So we mimic kind of the transfer from the mother to the egg. So normally the mother will put the pollutants inside the egg. Now we did it ourselves. So the first experiment focused on flame retardants. So like I mentioned before, flame retardants, they are used to make sure that we are fire safe in a way. So they retard in case of ignition, they will like make it slower, that things catch fire and the fire will start slower. So they are basically inserted in lots and lots of products not nowadays, because all synthetic polymers are highly flammable. So here you can just see some examples of products that the flame retardants are in. And there are also many different classes of flame retardants, and you can see many different structures. So my focus will be on emerging and re-emerging flame retardants, so not the ones that are regulated. And so one of those re-emerging classes of flame retardants are organophosphorus flame retardants, because they were actually already produced early on, maybe in 1960s, 1970s, but they were actually been replacing the PBDEs that were banned in the Stockholm Convention, and then they have been using them a lot more. And one of those compounds is TDCIPP, of which the structure is given here. So it's, let's see that there is like a phosphorus structure, but there are also chlorides on the structure. So this is one compound that we used. And the other compound that we used is dichlorane plus, which is actually proposed for listing as a POP. And it's also, you see, a highly chlorinated compound, but does not have a phosphorus, and it has a completely different structure. So the experiment, what we did is we made different groups. So we injected the eggs, like you can see here, with a needle. Need to be very careful, and then you have to kind of seal the hole so that it cannot get bacterial or viral infections in there. And so we had different exposure groups of dichlorane plus at 3 different concentrations, of TDCIPP, 3 different concentrations, and then we made a binary mixture of the 2 at the same, like 50-50. And we had two control groups, basically to control for the injection and also the emulsion that we used. And then we incubated them in this like incubators where you have this automatic rolling of the eggs. And then after some time, you get this cute little chick. So what I will present, because we looked at more things in this experiment, but what I will present is basically during the egg incubation, we took out one egg a day, and that you can see here. And in these eggs, we were measuring the compound TDC-IPP and the possible metabolite BDC-IPP. So I will show you. So BDC-IPP is basically where there is one of these missing. So it's like cut off, basically. So what we can see here is that over time, suddenly the concentrations of TDC-IPP start declining and those of BDC-IPP start increasing. And this is in fact the timing when the embryo starts to gain some metabolic capacity. So here you just have an egg and then you have like more developed embryo. So and you can see that by the end of the experiment, all the TDC-IPP in the egg was gone. And the BDC-IPP was increasing, but it was not increasing at the same level as the TDC-IPP, which made us think that probably it would be even metabolized more. But when we were looking for other metabolites, we couldn't really find them. When we then go to dechlorine plus, we can see a completely different pattern. So we also took egg out every day of the incubation of the eggs, but it basically stayed the same. So there was no change in the concentration. And this is like the injected dose. So it's kind of fluctuating around that injected dose, what we find. So there seems to be no metabolism of dechlorine plus inside the embryo. So this already points to us that emerging contaminants, these very different compounds. So both are flame retardants, but they have a completely different structure and they also can be differently metabolized by the avian embryo. So then we go to the second experiment. And that experiment is on perfluorinated chemicals, or PFAS. And they have very diverse structures, but here one structure is shown. So that is like in general, they have like an hydrophobic tail and they have a functional group at the end, which is more hydrophilic. So they have amphipathic properties, meaning that they can both repel water and fat. And they have, because of these properties, they are very much used because the consumers like this type of properties. They like these Gore-Tex jackets that kind of protect us from the rain. They like the anti-stick pants. This is, for example, ski waxes, or also for protecting like leather in your car and things like that. They are used in lots of products. And also this is more concerning in also many food contact materials, like these popcorn bags, or maybe also in the paper cups that you have your coffee in. So they are used in many, many products. And there is a very large variety of perfluorinated chemicals. So this is not even covering many of it, but this is, for example, PFOS, which is the regulated one. And then there are similar compounds, just adding every time a carbon atom. So the chain can be longer or it can be shorter. But then you can see that industry is also very much inventive, and they make also like this kind of combinations of compounds, and those are not at all regulated, right? So they make just different types of combinations. Then we have also compounds that we can call pre-FOS, because they're kind of precursors that can be biotransformed to PFAS in the environment. And then we have also the fluorotelomere sulfonates, like these ones here. So they are just a bit different. And then we have both the sulfonates and the carboxylic acid. So this one is the PFOA. So there are many different PFCs around, and this is not even covering it. So what we wanted to do in our experiment is to look at the difference, or if there was any difference, basically, between PFOS, which is the regulated compound. It's already included in the Stockholm Convention. And then we wanted to look at an emerging PFAS compound, and this one was F53B. So what you can see it here, so it has a bit different structure because here is a chlorine atom at the end, and there is also an oxygen here in the chain. So this compound is not at all regulated. So, and what we did is we again used different doses, but this time we used only 3 doses. We used 2 doses. We used a low dose and then 10 times higher dose. And we injected that again in the eggs, only PFOS, only F53B, or a 1-1 mixture of the two. And then we looked again at what is happening inside the egg and with the embryo. Also, this one, it's published, so you can read those articles if you want to know more. So what we found is that we looked at the accumulation of the compounds inside the liver of the embryo, and we found 15 to 19% of the injected PFOS in the liver. So it's quite a high concentration because that's only the liver. For F53B, we found 10 to 16% of the injected dose in the liver. So it seems that both are accumulating quite well in the embryo. So we do not see any indication that there is a biotransformation going on here. We also looked at effects of the exposure to those compounds. And we looked at different types of effects. We also look at oxidative stress and other parameters, but I show you only the clear effects that we found. And so we found an effect on the hepatosomatic index. And this means the size of the liver. So what we found is that in the control group, we have approximately this liver size. Then this is in the low dose, and then this is in the high dose. So we found, in fact, that in the high dose exposure to F53B, so the emerging compound, the liver was enlarged. So, and this can be an indication of that the liver is trying to cope with some intoxication. We don't know if this is like a toxic response yet. So it can just be adaptive, that the liver is more active to kind of cope with the contamination. We don't know if it's also a toxic response. But so important is we find this with F53B, but we didn't find it with PFOS. And then when we look at the embryo before hatching, we were monitoring the embryonic heart rate. So you have a nice device where you can kind of see just before hatching what is the heart rate. And what we found is that the embryonic heart rate was lower, again, with this high dose of F53B. We also found something in the PFOS low dose, but we didn't find anything in the mixtures. So there seemed to be no effect on the mixture toxicity, mostly on the F53B high dose. So this was important for us because it showed that the emerging compounds can actually seem to be quite toxic and can be more toxic even than the original compound. So this needs to be followed up. So what we try to do next, or parallel, let's say, is look at different contaminants in the raptor species in the Norwegian environment and see if we can find some of these emerging contaminants in them. And also we try to monitor and also look at different environmental factors that might affect their exposure and effects to these contaminants. So on this slide, I show you the locations that we had. So we had a gradient for the northern goshawk. So these are nestlings of the northern goshawk, which is a terrestrial raptor. And we had a gradient from Murcia in Spain to Trondheim in the middle of Norway, where I am based now, until Arctic Norway, Tromsø. So this species was monitored at these 3 locations. And then we had a more marine predator, the white-tailed eagle, and those we monitored at 2 locations, in Steigen, so more in the north, and in Smøla, more at the same level as Trondheim. And what is shown on this graph to the next is basically how they can differ in their diets, because we can monitor and study the diet of birds, but also other animals, using stable isotope ratios, so of carbon and nitrogen. So for example, here you have the ratio of carbon 13 over carbon 12, and here the ratio of nitrogen 15 over nitrogen 14. So, and what do these values tell you? Well, basically this ratio of the carbon, if it's like less than 20, let's say, or 22, it's a more marine diet. And this one would be a more terrestrial diet. So luckily, these are the northern goshawks, so they seem to reflect a more terrestrial diet. Although the green ones are a bit more to the right, so they seem to feed also on other species. And then you have here the white-tailed eagle, who seems to be more marine. On the nitrogen 15 axis, it means the trophic level, so how high they are in the food chain. But this also depends a bit on the baseline of the food chain. But you can see that that the white-tailed eagles seem to be on a higher level than the northern goshawks, who were basically in the same type of. There is variation within location, but not between the different locations in a way. So we took those values of their diet into account to look to the exposure. But first, I will show you the general contamination trends that we found. And so this is the concentrations in the blood plasma of the white-tailed eagle and the northern goshawk in the different locations, Tromsø, Trondheim, Murcia in Spain, Stege and Smøla. And what is very obvious from this graph is that PFAS seem to be the most abundant compounds in all of the locations. The red ones are the PCBs and the green ones are DDT and DDE, other compounds. And then you have some chlordanes, PBDEs, hexachlorobenzene, HEHs, and OPFRs. So, but they only seem to be minor contributors to the total contamination in the blood plasma. PFAS seem to be by far the most important compounds in all those birds. So that was very important for us. What we can also see is that the concentrations in Trondheim for the northern goshawk seem to be the highest. And this is mostly due to these higher PFAS concentrations. And in Murcia, the contamination seems to be quite low. So this might be related to the fact that in Murcia, it's a very warm and dry climate. So they probably have not a lot of need for things like Gore-Tex clothing and outdoor gear. And also they are not using ski waxes like they do, for example, in Trondheim, where there's a lot of ski arenas. Then if we look at the white-tailed eagle, we see that they are even higher contaminated than the northern goshawk. But this might be related to their diet and their trophic level, as we have seen that they are higher on the food chain. So they might actually accumulate more. So then if we look at the different samples that we obtained, because as I said, raptors, they are protected species mostly, and we have to be careful what we sample. So what we used is different types of sample matrices. We used blood plasma or whole blood. We used preen oil, which is like the preen gland is at the base of the tail, and it's where you see birds picking once in a while, and then they obtain some preen oil, which is spread over their feathers to protect them from water and dirt. So we also sampled some of this preen oil by squeezing the preen gland. And then we obtained some body feathers. And then we looked at the different classes of contaminants in these different matrices. And so the emerging ones are highlighted in red, and the legacy or regulated ones in blue. So what we found is that PFAS were dominant in the plasma. So that you can see here. Okay, there is also a higher concentration of PCBs and OCPs, so that's also high. But you see that the PFAS is absent, so we could not analyze it in the preen oil, and it's very, very low in the feathers. This, of course, preen oil is very lipophilic, very lipid tissue, so it has a higher concentration of the really legacy organic pollutants that are more lipophilic, like PCBs and OCPs, so the legacy POPs. While PFAS, they do not really like to be in the lipids so much, they like to associate more to proteins. So for example, like proteins that are in the blood. And then lastly, we found that the OPFRs, or the organophosphate flame retardants, that they were actually more abundant in feathers, while we could not detect them in the blood and we could not detect them in the preen oil. So this makes us wonder that probably this is an external contamination of PFRs on the surface of the feathers, because also as shown in our exposure study, these type of compounds, when they are inside the body, when they are taken up, they are quickly metabolized. So that's probably the reason why we couldn't detect any in the blood nor in the preen oil, because they are just not inside the body. But on the feather surface, they can be contaminated from the air, because concentrations of OPFRs in the air have been found to be quite high. So this actually shows that different type of sample matrices can tell you different stories. So it's very important to know the properties of the chemicals and also the properties of the matrix that you are using. If you are targeting lipophilic contaminants, you can sample the preen oil, but not if you're targeting PFAS. If you want to analyze for PFAS, it might be better to use blood. And if you want to know more about the, let's say, passive monitoring of PFRs in the environment, you can use PFRs. You can also find the other classes of contaminants in feathers, but they are at lower concentrations because, you know, feathers, they are not very lipid rich. But you can monitor them as well. So now we include some environmental factors into our analysis. And we were looking at the variation in plasma concentrations of different pollutants in the white-tailed eagle. And the factors that we took into account were the year of sampling, the location of sampling, the age of the nestlings, because we could, of course, not sample all nestlings exactly at the same age, because as I showed you, it's difficult to kind of take samples from raptors, and it takes a lot of time. And in the white-tailed eagle case, you have to go with a boat and you have to go from nest to nest. And also we monitored their diet. So, and this we did using the stable isotope values that we obtained. And what we found when using all these factors in our models is that we found the age of the nestling to be the most important predictor of the concentrations of contaminants in their body. And this is only nestling age. So this has nothing to do with differences over years between adults. This is just how old they are inside the nest. And the age ranged from 44 to 87 days old. So what you can see here is that you can see opposite trends of how the contaminants behave according to nestling age. So the concentration of PFAS in the plasma was higher when the nestling age was higher, while for PCBs and other POPs, the concentrations were declining with the nestling age. And this was also the case for the C13 values. They were also declining with the nestling age. So what we basically know that happens with POPs is that the concentration can be quite high in the egg because they are very lipophilic. So what may happen when the bird is growing is that this concentration, this initial concentration, is diluted with the bird growing. So that's why the concentrations are declining if they are not taking up a lot of these contaminants through the food. While with PFAS, it seems to be the opposite, because the PFAS in the environment and in the food are probably very high. So they are taking up PFAS continuously when they are growing. And then more importantly, we also looked at effects in those birds. And so this is very interesting because what you can see here is different types of physiological effects that we monitored in the white-tailed eagle. And for example, these are thyroid hormones. Then we have measured corticosterone in feathers, so the stress hormone. We have looked at total protein, different proteins like prealbumin, albumin, alpha globulins, beta globulin, gamma globulin, and also some blood chemical parameters that we measured. So, and what we wanted to know was the variation in our models, how much was explained of the variation that we found in different birds, how much of this variation is explained by the contaminants that we monitored. And this is given by this bar. So this black stripe in the different bars is actually the part of this variation that is explained by contaminants. And you can see that this is mostly quite low. This is like 10% of the variation explained. For some other, it may be a bit higher, but usually it's generally low. And that also kind of corresponds to the fact that the concentrations that we found in the white-tailed eagle were not skyrocket high. They were actually quite okay, and they seem to be in okay health also. So this is good. So the bird species is doing quite well in Norway actually, and also in Scandinavia, and they don't seem to have, for now, to have negative effects from the contaminants that they are exposed to. Although we should keep an eye on the PFAS contamination, because as we have seen, PFAS can be very potent, and new emerging PFAS compounds might have effects that we have not seen with the legacy ones. So then we go to the last part of this talk, which is about the interplay of pollutants and infectious disease in birds. So first, I want to explain to you the incentive for the research, why we started on this, this topic. And that was because I had a visiting postdoc, and she was doing a meta-analysis of studies on the interaction between chemicals and viruses. She had a background working on infectious disease, and she really wanted to look at this. And so when we looked in the literature, at that time, we found some exposure studies, of course, and there she looked at the overall control groups versus the treatment groups, and then she saw that there was usually a higher mortality with the ones that were exposed to pollutants. And then she also looked for the doses, so the different doses, both in vitro studies and in vivo studies, and she saw that in general, with an increasing dose to pollution, also the viral titer was increasing, so meaning that there seems to be more viral infection in those cases. So that was quite interesting, and that made us think about the following questions, like what is the prevalence of this in wild species and in wild birds, because those were controlled studies. And then we also wanted to know more about the mechanisms, how are pollutants interacting with the immune system. So the first part that we did was indeed going to the wild birds, and we did some sampling of black kittiwake, so a seabird, as you can see here, and we did that in Svalbard in Norway, so this really above the Arctic Circle. And there we took samples from 31 black kittiwakes. And the first observation that we made was that in 25% of the birds that we sampled, we find the prevalence of antibodies against avian influenza. And that was a first because avian influenza was not detected previously on Svalbard. So this was a very important finding in itself. And then we studied contamination in those birds, in the blood of those birds. We did both organic pollutants as well as metals. And so here I show you the example of arsenic. So when the concentration of arsenic were increasing, the probability of having had an infection, so having antibodies, also increased. So that is very important, and arsenic has also been reported to be mediating immunosuppression, for example, by reducing the macrophage activity in birds, and also it has been reported to increase the vulnerability to infection in mice. So, okay, I will show you an example later about an organic pollutant, but so this was something interesting. We found that also in wild birds, we could find these patterns. And so we wanted to investigate the mechanisms about this. How does pollution interact with the immune system? And so we looked at different biomarkers of the immune system. But we focus here on microRNA 155, which is actually this blue bar, because that's the one showing us effects. And so we exposed chicken cells, chicken fibroblasts, to a viral analog poly-IC. So we just used a viral analog because it has to do with health and safety issues. But so the viral analog will also behave and make the body think it is a virus, basically. And we also used Arochlor 1260 as a PCB mixture for the pollutant exposure. And then what you can see here is the healthy cells, so the ones that were not exposed to PCBs. And when we exposed them to the viral analog, they had an upregulation of the expression of microRNA 155. But when we exposed them to pollution first, and then we gave the viral analog, we had a decrease in the expression of the microRNA 155, which is very apparent. So this seems that microRNA 155 is involved in this process. And this is not to say that only microRNA 155 is involved, because there are many more microRNAs, and that is actually something that we are investigating now in our current project. Now, if we go back to the field, if we look at the expression of microRNA 155 also in the kittiwakes, and we compare that to the DDE concentration in their blood, we can also see a decrease. So the higher polluted the birds were in the Arctic, the lower the expression of microRNA 155. So again, confirming what we found in the lab. So in my current project, called Coast Impact, we look more into this interplay between pollution and disease, because you have all these shorebirds that are migrating from the Russian Arctic over the East Asian coast towards Australia. And so in this flyway, it has also been reported a lot of avian influenza disease. And we know that the birds populations for many of those species are declining. Some are declining very rapidly, others are more stable. So my collaborator in Australia, Marcel Claassen, has been investigating the influence of disease for a long time already. And he approached me and said, we have to look into the effect of pollution as well. And so we started investigating pollution at different stopover sites. Here you see them on the map in China. And there we collected sediment and mollusks that they may eat. And so we look at those different sites for the pollution. Again, this is a project with many partners. We had a startup meeting and also another workshop, and it was all over Zoom because of the pandemic, but nevertheless, we go ahead. And so we found POPs, so the legacy ones, and we monitored them in the sediment. And what you can see here is that mostly HBCD, so which is a flame retardant, hexabromocyclododecane, and also dichlorane plus and DDT. DDTs, especially in this location, seems to be quite abundant. Then if we go further, we see a different pattern in the shellfish. We see still like most contamination here also in the Bohai Sea, and some hotspots in other areas. But we see that this compound, tributyl phosphate, suddenly appears in the biota, suddenly appears in the shellfish, while we didn't measure it in the sediment. So this seems to be quite accumulating. And this is interesting because this is an organophosphorus compound also used as a flame retardant. We can also find some DDTs and some hexabromocyclododecane. And then if we look to more emerging compounds in shellfish, then we found that mostly parabens and PFAS are also very abundant. The same pattern was found in the sediment samples. So also this concentration here around Bohai Sea, but the concentrations that we found in the shellfish are higher. So they are accumulating as expected, these pollutants. So you see parabens and PFAS seem to be very important in those areas. Now, if we go back to the shorebirds, which basically is the main aim of the project, the collaborators in Australia, they have a lot of samples. So they have both blood and swab samples. And so they have already investigated also the avian influenza status. And you can see that this is two species, the red-necked stint and the ruddy turnstone. And this is basically the prevalence of the antibodies in those species. So almost 25% already has antibodies against avian influenza, meaning that they at some point in their life have had this disease. And then there was like a 5% in the ruddy turnstones that had actively a viral infection. So now if we go and look at the pollutant levels of the birds, so we measured them in their blood, then we can see for the different species, so this is the red-necked stint again, and this is the ruddy turnstone, and this is the curlew sandpiper, one other species. And so what is a bit sad about this is that we had a lot of samples of ruddy turnstones, but they were obtained from another area, King Island, and they seem very lowly contaminated. So we cannot really look at a pattern here because many are also below the limit of detection or close to the limit of detection. So then we found some interesting patterns for the other two species, but the problem is that we only have 2 and 5 birds that have had a positive status for avian influenza. So we are currently analyzing more of the redneck stints to get a higher sample size for the avian influenza positive versus negative ones. You see the red is negative and these ones have antibodies. So we hope to confirm very soon if this pattern holds. But so the ones that have had antibodies or have had an infection previously for avian influenza show to have a higher level of PFAS in their blood. So this is quite interesting. So what we are doing right now is working more on the link to the microRNAs. So we have already screened for many different microRNAs, and we are now investigating which microRNAs might be most predictive for these immunotoxic effects. So more updates are to come in this project, and you can also keep an eye on our website if you want. So that concludes my lecture. I want to thank all my collaborators and students that have worked in these projects, and also, of course, the birds that willingly and unwillingly have helped our research. And thank you for your attention.
Thank you so much, Professor Jaspers. So great presentation that covers so many interesting about birds, about pollutant, about even virus diseases. So now it's time for questions from the audience. I don't see any, well, I see one hands up. Maybe I just unmute this audience so you can talk.
Should I stop sharing my screen or?
I think it's okay.
Okay.
Just, yeah. It's I can't see the full name, but it's Xu XR. You can unmute yourself, I guess, and to ask a question. No. 唐老师，那个 chat 里面有一个好像举手了，你看看，您看看，就是让他问问题。
他好像又取消了。
OK。
取消了。哦，打出来了。嗯。
Okay, so the question is, great talk, thank you. I wonder whether the sample volume is a challenge for chemical analysis.
Yes, it is very much a challenge, especially with those shorebirds who are, some are very tiny and we can obtain very little blood from them. And that was great for working with raptors because there we could obtain much more blood, and we still have actually sample left from the new raptor project that we are still analyzing for different type of pollutants afterwards because we still have plasma left. But for the shorebirds, it's tiny, tiny volumes, and we luckily have good collaboration with the chemistry department here, and they are really optimizing their methods for making small volume detection for us. So yes, it is a real challenge, and we have to choose. That's why, for example, now we have focused more on PFAS in the blood of those shorebirds. If we want to analyze other type of pollutants like POPs, we have now obtained some carcasses, so we can actually in the dead birds look at the POPs, because it will be very difficult to analyze them in this small amount of sample.
Good. And I got another question from Simeon. So thanks for this great talk. I was wondering if there are other more general factors that could be correlated with the individual levels of pollution in shorebirds, other than the acute infections, such as immunocompetitors. Maybe that would increase that would increase sample size? That's a question.
Okay. Okay, so you're maybe thinking about looking more at general markers of the immune system instead of the active like viral infections or the antibodies, but you're looking more at the general immune system biomarkers. Is that what you are asking? I think so, but
yes, yes.
Okay. Yes, but yeah, that is indeed a good point, exactly. But the problem is also, again, I think that we have to prioritize what we can do with the blood. So what we can really look at in those birds, and we have to kind of prioritize to choose something, and if we do that, we cannot do the other. So, but thank you for this comment, because that can be interesting to look at in the future. Yeah.
Thank you. Is any more questions? Because I have many.
Okay, but you can go ahead if you want.
Okay.
Oh, there's someone else.
Yes. Well, Valerie, you can see it, right? So you can just read it from the questions and answer.
Yeah.
Maybe I will just read the question because audience from other platform cannot see this. So,
okay.
Yeah, so the question is, did you, of course, thank you for the talk, and did you measure other toxic endpoints than liver size, and what are changes you think can have important implication if you are using the chicken embryo model?
Yes. We did look at other endpoints. So we looked, for example, at expression of genes related to oxidative stress and antioxidant capacity. But we didn't find very clear relations there. But there is an article published on this. And you can go and check out everything that we wrote on this article. So Briels et al. I think it's 2019. So you can look for that one, and then you can read everything, because the most important effect that we found I have shown you. Yeah. And yes, it is also it's about the implications. Yes, yeah, I think of course this can have implications for the chicken embryo, because if for example the heart rate is not increasing enough before hatching, it can also have implications on the actual hatching process, because the birds they need to pip, they have to destroy also the eggshell, and they have to come out. And actually the endpoint of pipping and hatching is an endpoint in itself on how the birds are doing. So this can have effects. And also the enlarged liver, we don't know, because we don't know if it has at this point had any toxic effects, maybe not, but it's an indication that at least this compound is affecting the liver metabolism.
Thank you. And there is a following up question regarding, again, the limited sample size or the challenge of the sample size. So what about the fecal samples or poops? Can it be a good one, particularly for threatened species?
Yes, fecal samples have been used previously for monitoring contamination. And it is a bit dependent because the problem with fishes is it's also a dirty sample, so you need to do some cleanup there, but it can be used quite easily, I think, for metal contamination. One other thing that is a problem maybe is that what you measure in the fishes is what they are excreting, right? Not what they are accumulating, but it can tell you something about what they have been exposed to. But on the other hand, what you can find in the fishes can be the metabolized compounds and not the parent compounds that they have they are exposed to, because they excrete mostly the metabolites. So and some of the compounds that we maybe are targeting are maybe very readily metabolized, and maybe then you don't find anything and you don't know what they are exposed to. But yes, fishes can be an alternative for a sample for measuring pollutants, but I think it's mostly maybe useful for elemental pollution.
Thank you. And the next question is why the infected birds have the tendency of more accumulation of PFAS?
Yeah. Why? Yes. No, I think maybe, but this is just guessing, right? Because this is just something that we have done recently, and we need to investigate more. But I don't think that it can be related to each other. So what we try to investigate is to see also if the birds that are stopping over maybe at more polluted places, taking up more PFAS inside their body, if maybe their immune system is somehow compromised, so they are more easily infected. So that would be the hypothesis. We still have to prove the hypothesis.
Thank you. There is a question from the live streaming platform saying, well, it's in Chinese. So is there any measurement data on F53B concentration in the European natural environment? And how it, if it does, how it compares to other regions?
I don't know much literature on the F53B, but we started investigating this compound because it was found in the Chinese environment, as far as I recall, and it's used in the electroplating industry. So it has it has also an essential, let's say, more essential use, because I don't think they have many options there in this industry. I don't know if it's also used in other applications. That might well be because it's not regulated, right? So, but we started looking into this compound because it was reported more in Asia. As far as I know, it's not been much reported yet.
Thank you. And another question from the live streaming, well, is any study about the metabolism of 50B or PFOS in chicken or birds already?
Can you repeat that question?
Yeah, is there a study already looking at the metabolism of 53B and PFOS in chicken?
We didn't target specifically looking at the metabolism, but these compounds are very persistent. so as far as I know, there is no way PFOS is metabolized, because that would be that would be a breakthrough, I think. So at least not in wildlife. Maybe some bacteria can do it, but yeah, they are very persistent.
Yeah, thank you. And another question is about the microRNA. So do we have enough evidence to show that the microRNA 155 is related to immunotoxicity, and whether there's any like indicator or biomarker that can be used to indicate the virus infections in birds?
So these are two questions, right? But but the first question about the microRNA 155, well, it's difficult. So I didn't show that mechanism, but microRNA is just like a sub, how to say, it's one step in a cascade of events, right? So the microRNAs, what they are doing is they can regulate expression or translation of genes. So they can kind of bind to messenger RNA in a way, and then stop that this one is translated into proteins. So that's one, and different, of course, different microRNAs, because there are many. They can therefore also have different effects, because they have different sequence, basically. So they can kind of have effects on different type of proteins. So microRNA 155 has been related also to some human, so that's how it actually, we started on this. So in human cancer research and other medical things, they started looking into microRNAs as biomarkers of different type of disease. So, and microRNA 155 is one that is mentioned to be involved. Like I said, this is an ongoing research, and for us specifically to do this in birds, and it's not a given that all these microRNAs are similar in a way to what is happening in humans. So it has to be investigated, these different microRNAs, which ones might play a role and which might ones may not. And we did a whole microRNome basically, but we are still processing all the data there.
Thanks. And is there any more questions? I don't see more from the chat or the question and answer part. Maybe I should take the chance to ask a question. So it's pretty scary to see the white birds are so contaminated, although they have the ability to still survive. But I'm wondering, you also use chicken embryos as a model. So what about the domestic chicken we eat? Did you have any data or thought on that? Or if they are also contaminated, what about human health?
Yes. Yeah. Yes, it is.
Yeah.
It is in fact a real issue. And I don't know if you hear about this, but I originally come from Belgium. And in Belgium, you have this factory 3M, and it's producing a lot of PFAS related compounds. It has been doing that for years. And of course, when it phased out PFOS production, and before also, I think, and already maybe in 2000. But there is a lot of legacy contamination in that area. And recently it came out in the media. We knew this already, but that these areas are heavily contaminated around the city of Antwerp, and people have been consuming their home, like chickens, for homegrown chicken eggs, the vegetables in their gardens. And now, of course, they have put all these regulations in place that people should, or recommendations in place that people should not do that. So yes, all these compounds that are in the environment, they get into your chickens. So if you have chickens at home and they are going around in a close to, I don't know, a contaminated area, or maybe agriculture even, or everything is transferred to the chicken egg. And it's actually better to have chickens inside than outside, because they pick everything up from the ground and they are more contaminated. Yeah.
Thank you. I think we are almost time. Now it's 10. We have a great 1 hour already. Time flies. 唐老师，我们现在结束吗？还是不是时间到了？
可以结束。不过这边那个 Q&A 就是问答那边有3个问题，您还看到了？是不是已经回答过？就是在问答那个有4。
Okay, I think there is one last question from the Zoom. Is it possible to estimate the accumulative PFAS concentration via human consumption of chicken products?
Yes, I think so, because there are some, they do these food basket studies, and they have these daily estimates of intake. So based on what people consume, for example, do you consume 1 egg a day or 2 eggs a day, or per week? Maybe not per day, but it depends. I don't know what people are consuming, but they can make these estimations of how would be your total weekly intake, for example, based on your diet. And then you can start estimating how much you take in over a week or over time, and then you can start estimating how much of it will stay inside your body. So people are doing that.
Thank you so much, Professor Jaspers. So I think we should just thank you once again to give the time for our journal and to the audience. And with that, we will just end today's talk at EEH. Thanks a lot. Thank you, audience.
Thank you very much.
Thank you very much. Thank you, Professor Jaspers.
Bye-bye.
Bye.